University, Ondo, Nigeria. He's also a research and development consultant and an academic editor for respective biotechnology journals. Please.
Ladies and gentlemen, colleagues, good morning. The topic before me is the incidence of extended spectrum beta-lactamase producing bacteria in salad vegetables in Ondo State, Nigeria. The preview really is that we are aware that there's no sterile environment and there are always potential pathogens in food products, and salad vegetables are not left out. We are aware that because of the nature of salad vegetables, because they are consumed raw, most of the cases salad vegetables could either be consumed raw or cooked. But most of the cases we have them raw, and because of this we are prone to consuming potential pathogens. We do not really think in this direction because we have immune system. We believe we are so immunocompetent and we don't fall sick as often as we ingest these potential pathogens. Also, other factors could also be that probably because of the pathogenicity of the potential pathogen. Of course, there's a degree of pathogenicity maybe that's referred to as virulence. So it depends on the virulence of the organism that we ingest every time, or the pathogenicity or the immune competency that determines whether we are falling sick as a result of the pathogen we consume. However, it is no news that we consume pathogen every time. But the problem is the development of resistance to the antibiotics that we use. We have used a wide array of antibiotics. We have carried out a series of antibiotic sensitivity testing, and we realized that at the end of the day, most of those bacteria that were one time susceptible to a set of antibiotics are becoming resistant. The major aim of this work is just to channel research to this direction, because extended spectrum beta-lactamases or extended spectrum beta-lactamase producing bacteria have not just been isolated, but it has been channeled in the past towards clinical samples. Clinical samples, maybe urine, blood, contaminated blood, cerebrospinal fluid and all that. However, we do not actually look into the possibility of having vegetable salads as vehicle of ESBL producers. Food safety begins at the farm. It's not usually at the grocery or in the kitchen. Fresh vegetables are considered as the essential components of every diet of people, and consumption of vegetables in the form of salads has increased in many parts of the world, and of course, Africa is not left out. They are major reservoirs of opportunistic and emerging pathogens due to its diverse microbiome, strongly influenced by geographical aspects of farming and food processing practices. Another thing is even that the soil itself, where we have all these produces, is a complex microbial habitat. The most complex microbial habitat is the soil, and we can find virtually all microorganisms in it. And because of that, I believe that there are still thousands of microbial species that are yet to be identified or cultured. So in contrast to the potential health benefits of fresh vegetables, concerns have been raised about safety and quality of vegetables due to outbreaks of infectious disease. Fresh vegetables are consumed raw, just like I said, and that makes it potential risk The non-pathogenic microbes associated with plants as commensal pathogen may lead to allergies that are undeterminable. Yes. No. So, the major concern here is the resistance to pathogenic organisms to countenance antibiotics. You know, it has become a worldwide problem, and we are looking for a way to improving that. And I was wondering, the first lecture that came was actually moving towards that direction. The second one also, partly on food safety and all that. I feel, the previous speakers are even giving me a soft landing. So the heightened use and or misuse of antibiotics in human medicine, agriculture, and veterinary is primarily contributing to this development of resistance that we are facing, and it's a continual thing. It is a continual thing as far as man lives, as far as industrialization continues, as far as we make use of antibiotics, we abuse drugs, then antibiotics become weakened in nature. And that is why here we have the mutant emerging here. These mutants emerging slowly. At the end of the day, what we are facing really is a matter of survival of the fittest. Because at the end of the day, the sensitive cells will die of those ones that are susceptible to antibiotics. They will die, the mutant remains, and at the end of the day, the mutants proliferate and they take over. There are different classes of antibiotics. This is just an overview. However, I am interested, and this study is interested in evaluating the beta-lactams. The beta-lactam is an enzyme deactivating the beta-lactam ring present in those beta-lactam antibiotics family. The beta-lactam ring is part of the structure of the beta-lactam antibiotics. It is a major structure. It contains the beta-lactam ring in their molecular structures. So nitrogen, this is the ring after which the name beta-lactam is brought about. Nitrogen is attached to their carbon relative to the carbonyl ring and hence that name. So most of the families of the beta-lactams, beta-lactam antibiotics, the penicillins, the cephalosporins, the cefamycins, carbapenems, monobactams, these are just examples, the representatives of those antibiotics. Extended spectrum beta-lactamases are plasmid-mediated. The genes encoding these enzymes could be transferred easily among bacteria. We know that plasmids are actually extra chromosomal genetic elements that confer special characteristics on the bacterium, on the host bacterium that possesses it. And those special characteristics could be pathogenicity, virulence, antibiotic resistance. And in this case, it becomes very important because the ESBs are even plasmid mediated, and transferring them from one organism to another is so easy. ESBL hydrolyze oxyamino cephalosporins and transmit resistance to bacteria against penicillins, cephalosporins. Those are the first to third generations, and to astrina. These resistance to the first to third generations could be repressed by beta-lactamase inhibitors, and we'll come to that. ESBOs are classified as beta-lactamases containing 3 main families, the 10, named after the patient. Thermonaria, the SHV is of hydrogen variable, and CTXM active on cefotaxime, first associated immunase. Most of these plasmids not only contain DNA encoding ESBOs, but also carry genes conferring resistance to several non-beta-lactamase beta-lactam antibiotics. Now, the presence of ESBL in bacterial isolates has been documented as a very serious problem because it brings about risk to quick survival of patients in hospital. It makes treatment so difficult. And as a result of that, it brings about high economic burden, and by extension, loss of honors, loss of hours, loss of hours in life activities, and huge treatment failure. Justification of the study here. So why is this study necessary? In spite of potential beneficial aspects of salad vegetables, concerns over their safety and quality have risen. Salad have little or no processing steps that can reduce the level of pathogens. The presence of bacterial population as endophytes cannot be removed by the simple washing, and thus enters human during consumption of fresh produce as raw salads. There were several reported outbreaks related to salad vegetables in the past, but none has actually focused on ESBL producing bacteria in southwestern Nigeria. So reports of Fodi et al., Andrew et al., Nepa et al., Jose et al., all of these are actually channeled towards targeting ESBLs from clinical samples. So this study is to investigate ESBL production in salad vegetables. So is it a simple experiment channeled towards neglected area? Simply, we made use of cucumber, carrot, green pea, green beans, sweet corn, which were collected randomly because it has to be measured. 25 grams were measured and blended in a 2 to 5 milk per ton. Resolution was carried out to up to 10 raised to the power 6. The nutrient agar was actually employed to determine the total viable bacterial count, McConkey agar for the total coliform count, the EOC methylene blue for Escherichia coli, and at the same time fecal coliform count. So mannitol salt agar is specific, is a selective medium for Staphylococcus aureus, and which was also employed. So the pure isolates were obtained and characterized accordingly. It is supposed to be a simple experiment, really, where normally after this stage, ground staining spore and the basic biochemical test, they should be able to identify primarily those organisms, the potential pathogens present in the food. But I decided to confirm, just to be double sure, and made use of the API 20E and API non-20 to confirm the presence of the Enterobacter C and the non-Enterobacter C respectively, because it happened to me once when I had my isolates characterized primarily like that, and I was to identify those organisms. Tentatively, I took the isolates to a lab in South Africa, where at the end of the day I found that most of those organisms were not even what I identified initially. But now it will be fine if basically one can confirm with the API. Now, for the inoculum standard, the turbidity was prepared up to 0.5 Mafala standard. Then, for the breakpoint of ESBL screening now, the inocular isolates, we made use of the cefotaxime and ceftazidime, really. Primarily, this is a double synergy test, double disc synergy test, and any organism that is resistant to any one of these two members of the cephalosporin is assumed to be a potential ESBL producer. So, and the method of Kebiboa 1966 was used, interpretation was done by the Clinical Laboratory Institute. This is the confirmation test. The isolate, they were further inoculated on MHA, that's Melaitin agar. So now the difference between this confirmation test and the previous one is that initially we will have the inhibition, the source of inhibition. And here we are not making use, previously we didn't make use of the amoxicillin, the clavulanic acid, that is augmentin. So for the confirmation now, if peradventure at the introduction of the augmentin, we now have enhanced zone of inhibition, enhanced diameter. as a result of introduction of augmentin, then the isolates giving such responses, and peradventure that now have zone of inhibition greater than 25 for cefotaxime and 22 for ceftazidime. They are confirmed as ESBL producers. So the statistical analysis was also carried out, and the MEDCAC is not really a popular one, I don't know. The MEDCAC is a statistical software package designed for medical and biological sciences, and the one-way analysis was employed and the independent t-test. Now, this table shows the quality of vegetable salad sold in Nigeria. The TVBCs, as total value by their count, range from 1.1 to 10 to the power 3 to 7.1 10 to the power, I'm so sorry. So, and the TCC, this is the lowest range, and we have this as the highest. FCC range from 0, to 2.9 times 10 to the power 2 colony-forming units per gram. So the mean of this, the mean TCC was highest in cucumber and lowest in green pea. The mean TCC was highest in cabbage and carrot samples with 2.8 times 10 to the power 3 colony-forming units, that's per gram. While the green pea had the lowest with 1.3 times 10 to the power 2 colony-forming units per gram, and this is the table showing the results. Okay. There was radical differences in mean TVBCs of the samples, and with significant value less than 0.05. The mean TCs of cabbage, carrots, and cucumber showed no statistical difference. Green peas, green pea, and sweet corn also showed no statistical difference. The mean FCC of cabbage was statistical difference from other samples. Table 2, that's just the morphological characteristics and the biochemical characteristics of the isolates. And here, we were able to isolate 166. We obtained 166 isolates, and they were characterized into 9 genera, about 10 species. And this includes the Bacillus cereus, the Citrobacter frundi, Escherichia coli, Klebsiella, Morganella morgani, Pseudomonas aeruginosa, Proteus mirabilis, Staphylococcus aureus, Serratia marcescens, and Staphylococcus saprophyticus. So this is a table, and these isolates, we are able to obtain this number of isolates this organism. So the distribution of bacteria in salad vegetable samples was shown in the next table. The essence of this is just to see which of these salad vegetables actually contributed to the microbial diversity that we have encountered in this study. And this is it, cabbage, carrot, and all that. So they were there, and they all contributed in a way to the microbial diversity of the samples. This figure shows the percentage occurrence of bacteria isolated from salad vegetables. S. aureus are the highest, followed by Pseudomonas aeruginosa, Pneumoniae, S. marcescens, B. cereus, and Bacillus, Proteus mirabilis, Morganella morgani, S. saprophyticus, Escherichia coli, and C. frundi. And this, that's the illustration. Now, this table shows the screening for potential ICF producers based on the CLSI breakpoint using the two cephalosporins family and the double disc synergy test. Here, these are the isolates. But for the first preliminary test, we were able to have the potential ESBL producers out of the 6. Out of the 16, we have 6. And after the confirmatory test, we have 4. So that makes it, that makes the ESBL producer 4. We have 4 Bacillus cereus out of 16, 1 out of 7, 4, 9, and like that. The table illustrating that, the figure illustrating that will be shown after this. So this is it. At the end of it all, we had Bacillus cereus and Escherichia coli as the highest ESBL producers with percentage occurrence of 2.41, and followed by Pseudomonas aeruginosa and S. aureus, Morganella morgani, and Klebsiella pneumoniae. The lowest are the C. frundi and P. Proteus mirabilis and Serratia marcescens, where Saprophyticus is not just an ESBL producer. Discussion. The organisms isolated here are majorly from indeed, they are contaminants of soil. And just like I said, soil is the most complex microbial habitat, and people have escaped to the produce during farming process, irrigation water, and all that. The high microbial contamination observed might be as a result of the storage conditions also after obtained from the farm, then handling, and the likes. So the total viable bacterial counts obtained in this study were lower than those reported by Kaneko et al. 2013. And Cantwell also reported that Aedes microorganisms related to eat vegetables could be due to plenty of cold surfaces, which allowed increased nutrient availability and thus favor the microbiota associated with the fresh products. The low level of microbial loads in green peas, because that's why we have the lowest microbes, could be as a result of the fact that green peas were protected in a pot and thus prevented them from direct contamination, while that of green beans could be as a result of their smooth surface and their protective manner of display, even at point of sale. Nietzsche and Babu also reported similar. They reported some organisms, Stretronophomas, Azophila, Atrobacter, Mysorens, Xanthomonas, Actinopodes, and Aeromonas hydrophilia. But all these were not encountered in my study. And my study also differs from the study of Kemajo et al., who reported Escherichia coli as the predominant organism, and followed by Staph aureus in vegetable leaves. Enterobacter aerogenes and Salmonella species were not also encountered in my study as reported by the authors. So, Enterobacter species, Escherichia coli, Klebsiella, found in the sample were actually contaminants. Were actually contaminated with matters. It indicated that the samples were contaminated with matters of fecal origin, with feces, with the soil sewage, and probably as a result of the poor quality of water used during the process. The presence of S. aureus could be due to poor hygienic practices, and of course we know that it is in normal flora and is carried in another passage of food handlers or infected workers who aid easy distribution of the organism. So most strains of Staphylococcus aureus could produce its stable heterotoxin and thus are antibiotic resistant and pathogenic in nature. I've also carried out the previous work on that, where it was reported that improper handling and poor hygiene might lead to contamination of food and thus pose serious health effects on consumers. So the presence of ESB producing microorganisms in salad vegetables may contribute to horizontal spreading of resistances between different isolates, species, and genera. Bacteria serving as a reservoir for resistant determinants may have great influence on resistant gene transfer in natural habitats, such as vegetal surfaces or human colon. So therefore, the presence of antibiotic resistance in fresh salads constitutes an additional concern for human safety. Conclusion. This study revealed that salad vegetables be a vehicle for the spread of ESB-producing bacteria, which translates to a threat to public health around the world, as salads are loved and consumed by all categories of people globally. So I have some recommendations here. These are my references. Thank you for listening.
So a question, basically it drives, let's say the recommendations, I didn't have time to see it, but it could be then, let's say I assume that we cook all our vegetables and then we keep those 21 isolates from the 166 that shows resistance against the bacillolactamase antibiotic. But then that means that the plasmidic fragment that confers resistance to that microbe, actually the microbe is killed. But we know that even by boiling, the DNA is still there. So now let's infer that we eat the vegetable. And then in the microbial gut, that DNA would be not recycling and confer resistance or not? What is the scenario after cooking? What do you think about it?
Well, I mentioned that briefly earlier. Many a time, it's not even about cooking. It's just this one is just emphasizing the fact that the raw vegetables could pose a greater threat. Even those food that we cook. No, of course, microbes are ubiquitous, and no material is sterile. So we ingest microbes virtually every time, even the cooked ones. As a matter of fact, during the process of eating, you cannot eat a bowl of maybe rice in 1-2 minutes. So in the process of eating, maybe overstretched 5 to 10 minutes, you are eating, microbes are in the atmosphere transiently. They are settling on the food, even that you believe that you have cooked very well, you ingest. So we are just creating awareness that because we keep talking about antibiotic resistance. The best way to go about it is just to block all possible ways where all these organisms will be spread, and of course where the genes will be transferred from one organism to the other, the resistant genes. So in the process of consuming contaminated food, even cooked ones, they are contaminated, you ingest them, and that was why I mentioned that we don't fall sick as often as we take all these food products, because most of the cases we are immunocompetent. The immune system, if the immune system is, we have self-defense, and except that due to stress or some other underlying factors, the immune system is tampered with and will now become immunosuppressive, and coupled with the fact that the nature of the potential pathogens, you do not know the pathogenicity or the virulence of the organism, so those ones are factors too.
All right, thanks.
So does the microbial community present in the salad, can it depend on the location from where you have collected the samples?
Yes. Well, it could be a factor, because that was why I mentioned those in Nigeria. Yeah. But generally, and microbiologically, you would agree with me also that your soil in your country is the most complex microbial habitat. So also in Nigeria. So if that happens, if it's the most complex, you find all sorts of potential pathogens in there. We find all sorts of potential pathogens in our soil, and it grows from there. You package, and due to some carelessness too, hygiene practice and all that, all these are factors everywhere. So it wasn't. I could even infer, I could conclude that we could encounter this everywhere. since it's coming from the soil. Yes, sir?
Thank you. The last sentence, to combine beta-lactam antibiotics with a drug that disables the resistance mechanisms.
The second one?
The last sentence.
Okay.
Yeah.
Okay.
What do you mean by the disables the resistance mechanisms? Do we have antibiotic disable the resistance?
What that means really is that
Yeah.
to combat the problem of the beta-lactam, the extended beta-lactam means producing bacteria. The problem could be achieved, it could be overcome rather by designing new ones. Then two, combine current beta-lactam antibiotics with a new drug. Which will now disable the resistant mechanisms. The resistant mechanism could be a plasmid. The gene responsible for
plasmid mediated?
Yes. So
plasmid mediated?
Of course. PSBS are plasmid mediated.
Yeah.
So the mechanism responsible now for the transfer of the resistant genes, and because the special characteristics have been conferred on the resistant bacteria by the presence of the plasmid. Definitely, if the plasmid is the problem that carries the gene, if the plasmid carries the gene, it could be destroyed. So that if it can disable it, then it will be achieved. Just the combination of the new one with the current one. That is what I mean.
Yeah, thank you.
Thank you, sir. Yes, sir?
I actually have no question, but I know it. So I noticed that during in all your presentation, basically, you have different ways to write the Serratia marquescens.
Serratia marquescens?
Yes, and none of them is the good one. So
I'm sorry about that. Probably I didn't notice. Serratia marquesens.
It should be with the S and the C.
Okay.
And you write it with a C, and the one time you write it with the S, just to let you know.
Okay. Thank you very much.
Thank you.
Now we'll welcome Dr. Ashish Badi. He's from Energy and Resources Institute from
I hope it didn't stop.
No, didn't stop.
Thank you.
But this direction was not proper, so I corrected it.
Thanks a lot.
Welcome.